invite Jamie Dean. Jamie Dean joined us recently to lead computational radiation biology.
Great. Thanks, Gary. So today I'm going to give you hopefully a little flavour of some of the areas I'm interested in working on. So I recently started as a junior group leader based at UCL, and so there are three main areas that I'm interested in. So firstly is understanding mechanisms of treatment response in order to identify new therapeutic strategies. Second area is improving the way we combine novel drugs with radiotherapy through optimizing administration schedules. And then thirdly, seeing if we can come up with better ways to personalize treatment in order to enhance the efficacy of radiotherapy and also reduce side effects. So I'm going to go through each of these areas in turn and give a couple of very brief examples of kind of work along these lines. So starting off with identifying new therapeutic strategies. The lab where I did my postdoc had done this very interesting study shortly before I joined, where they'd looked at trying to improve the way we administer radiotherapy for glioblastoma. So current treatment approaches don't account for the heterogeneity and plasticity that we know are key features of glioblastoma. And so they've done this study looking at whether you could explicitly model these different cell states and transitions between them in order to inform improved treatment approaches. And so they developed this mathematical model, a schematic is shown here, and used it to optimize a treatment schedule and validated the optimized schedule in a mouse trial. So in red, you can see the survival of a genetically engineered mouse model of glioblastoma treated with a standard radiotherapy schedule, giving radiation once per day. And in green, you can see the survival from the optimized schedule, which involved giving multiple doses of radiation a day with a specific time interval between the doses. And so you can see that led to a pretty large improvement in survival, and the magnitude of the benefit was not far worse from a doubling of the total dose using the standard schedule. So then I worked on translating this approach from mice to humans and doing a bit of extra statistical modeling to see whether the effect was likely to be beneficial in humans, coming up with a human appropriate schedule which also accounted for toxicity, and then designing a phase 1 trial in order to make sure this approach was both feasible and safe to administer to humans, which we were able to successfully demonstrate. A slightly different level, another area I'm interested in is non-genetic heterogeneity in radiation response, and how transcription factor dynamics and heterogeneity in transcription factor dynamics can influence cell fates. So as part of some work on that, I conducted a study which was looking at P21 transcription in response to radiotherapy. So here's a short video showing some live cell imaging. So on the top, you can see the dynamics of P53 over time in response to radiotherapy. And on the bottom, you can see these, this is imaging P21 transcription. So you see these bursts of P21 transcription occurring under these pulses of P53. So one thing I did was to come up with a mathematical model to try and describe the relationship between P53 and P21 transcription. And interestingly, a kind of standard model whereby the P21 transcription is just dependent on the concentration of P53, which binds and unbinds from the P21 promoter, wasn't able to fit the data very well. So here you can see an example cell where in gray you can see the P53 pulses following radiation. In black you can see the P21 transcription, and in blue you can see an example of a single instantiation of a stochastic mathematical model of the P21 transcription. And then here on the right hand side you can see this is how we compare the model simulation to the data through looking at correlation functions. And the important thing is that the blue lines don't match the black lines very well. So then I looked at another model, which is a network architecture called an incoherent feed-forward loop, whereby p53 also induces the expression of some kind of repressor molecule that blocks p21 transcription. That kind of model is able to describe the data very well. So this shows an example of how using mathematical modeling could lead to novel biological insights that might not have otherwise have been made. We did a lot of follow-up work identifying this repressor, looking at its role in other p53 transcriptional target genes as well, and then we're able to show that through manipulating the interaction between the repressor and p53 using a drug, we're able to steer the cells towards a temporary cell cycle arrest. So there's the potential to use this kind of approach for radio protection in the context of treating p53 mutant tumors. Okay, so the next area is optimizing schedules for drug radiation combinations. I won't go into too much detail for time, but just to give you a kind of idea of the approach, is to take longitudinal data of preclinical models treated with radiotherapy, drug, and then the combination. Use those data to fit and compare different mathematical models and find the one that's able to best fit the data, accounting for model complexity, and then use that model to design a preclinical validation experiment to test our predicted optimal schedules, and if they're successful, taking those through to inform early phase trial design. And so we've got some ongoing work looking at different DNA damage response inhibitors in combination with radiation at the moment. And then the final area is different ways of personalizing radiotherapy. So one thing we're working on is with being able to take advantage of new technologies like proton beam therapy, which are able to drastically reduce the irradiation of normal tissues. And so one problem we have at the moment is trying to understand which areas of normal tissue would be the most beneficial to spare. And so this is particularly important in treatment of pediatric tumors. And so we're doing some work on this, trying to understand which regions would be most beneficial to spare in order to reduce side effects. And then another area is using longitudinal imaging data from patients treated with radiotherapy in order to come up with personalized and adaptive treatment protocols. So that might look something like this, where we have some baseline imaging, which can be used to predict how a patient is going to respond either to the standard course of treatment, but also to design an optimal treatment strategy for that patient based on the data we have at baseline. And then as more imaging data are collected during the course of treatment, we can then update those predictions over time. So both being able to update how the patient's likely to respond to the current standard treatment protocol, but also how we might update a personalized treatment protocol over time as more data come in as the patient's receiving treatment. So hopefully that gives you a little flavor, if not too much detail, about the kind of things we're interested in. We're always very excited about collaborating with colleagues in the wet lab and also clinicians as well. And I'd just like to thank a kind of growing team of students who are getting a lot of this work going together with collaborators I haven't listed here because I haven't gone into our work in detail, but thanks for all their support as well.
Questions for Jamie? Okay, so I will start. Your oscillatory method, is that specific to GBM?
So we don't know yet. So the mechanism at the molecular level isn't understood, and that's something we're currently working on trying to understand. So once we hopefully understand what's going on the molecular level, then we'll have a better sense of whether it's going to be something something specific to glioblastoma, or specific just to this one mouse model, or whether it's a more general effect that we can exploit in other situations as well.
Okay. Tony?
Do you think, other than transcription, you could have degradation of p53 as an oscillatory mechanism? Because someone showed ATR, ATM, and that in itself affect MDM2 as well.
Yeah. So yeah, just to say, for that project we were looking at not in how p53 oscillates, but rather how p53 transcribes its downstream target genes. But yeah, for sure, the reason you get oscillations in p53 is because of negative feedback through MDM2 regulated degradation. So yeah, whenever you have oscillations, you always need negative feedback to produce that. So by altering MDM2, you can also get rid of this negative feedback and cause like sustained increases in p53, which alters cell fates as well. So playing around with, once you understand how these transcription factor dynamics are controlled, you can do all sorts of cool stuff with manipulating that in order to influence cell fates. So yeah, that kind of stuff I'm very, very interested in.
Thank you.
Okay, we've got a question from Anna Wilkins. Thanks for a great talk. Wonder if you think there is potential benefit of interrogating biological parameters from the tumour samples, for example, chromosomal instability alongside the imaging data?
Yeah, for sure. So yeah, I gave the imaging as a kind of example of the kind of data where it's kind of easy to get longitudinal measurements, but definitely I think there'll be a huge amount of value in looking at biological samples in conjunction with that, and in tumours where it's possible to get longitudinal measurements. So with things like circulating tumour DNA or things like that, that would be also very interesting and I think quite valuable as well.
Thanks very much, Jamie.
Thanks.